Hi, it's Alex Zubarev for KCICU Primary Study Group. Today we will discuss drug metabolism or biotransformation. This topic appeared several times in short answer questions in the past and may show up in the future.
Drug biotransformation or metabolism is the chemical alteration of drugs in the body. The main site of drug metabolism in the body is the liver. Other sites include the gastrointestinal wall, plasma, lungs, and kidneys. The general principle of biotransformation is the metabolic conversion of drug molecules to more water-soluble metabolites that are more readily excreted by kidneys. In most cases, the metabolism of the drug results in its conversion to inactive metabolites that have little or no pharmacologic activity. For example, morphine is metabolized into inactive morphine-3 glucuronide. In some cases, the biotransformation of an active compound may lead to the formation of active metabolites that also have pharmacologic actions. For example, another morphine metabolite, morphine-6 glucuronide, has an even higher mu receptor affinity than morphine. A few compounds, also known as prodrugs, have no activity until they undergo metabolic activation. For example, clopidogrel is a prodrug and undergoes metabolic activation in the liver. Lethal synthesis, or suicide metabolism, is the biotransformation of a non-toxic or less toxic precursor into a toxic metabolite. For example, the metabolism of paracetamol into N-acetyl P-benzoquinoniamine, or NAPQI, or the metabolism of methanol into formaldehyde. There are two broad types of biotransformation, called Phase 1 and Phase 2. Phase 1 reactions result in the modification of the drug molecule via oxidation, reduction, or hydrolysis. Phase 1 reactions can be also classified into microsomal and non-microsomal reactions. Microsomal metabolism involves cytochrome P450 isoenzymes. These are major enzyme systems localized in the smooth endoplasmic reticulum or microsomal fraction of cells, predominantly in the liver, but also in the gastrointestinal tract, lungs, and kidneys. Microsomal reactions have an absolute requirement for molecular oxygen and reduced nicotinamide adenine dinucleotide phosphate. The enzymes of the cytochrome P450 system are classified into families and subfamilies by their degree of shared amino acid sequences. In addition, the subfamilies are further divided into isoforms. Families are labeled CYP1, CYP2, and so on. The subfamilies are labeled as CYP1A, CYP1B, and so on. And the isoforms are labeled as CYP1A1, CYP1A2, and so on. Many drugs are metabolized by more than one isozyme. For example, midazolam is metabolized by CYP3A4 and CYP3A5 enzymes. Not all Phase 1 reactions are catalyzed by the microsomal enzymes. Non-microsomal drug biotransformation includes reactions of hydrolysis, for example, various esterases and amidases. Monoamine oxidases catalyze the metabolism of endogenous amine neurotransmitters like dopamine, noradrenaline, and serotonin, as well as the metabolism of some exogenous substances like dobutamine or metaraminol. Alcohol metabolism is another example of non-microsomal reactions. Alcohols are metabolized to aldehydes and then to acids by dehydrogenases. Genetic polymorphisms exist for these reactions. Phase 2 reactions result in conjugation with endogenous compounds and are catalyzed by specific transferases. Phase 2 reactions may follow Phase 1 or occur as the primary reaction of biotransformation. Enzymes of Phase 2 reactions may be located in microsomes or the cytosol. There are several common types of conjugation reactions. Glucuronidation is a reaction of conjugation with glucuronide. Examples of drugs metabolized using this pathway include morphine and chloramphenicol. Similarly, reactions of acetylation introduce an acetyl functional compound, reactions of glutathione conjugation introduce a sulfhydryl group, sulfation introduces a sulfate group, and methylation introduces a methyl group to organic chemical compounds. Glutathione conjugation is the main and non-toxic biotransformation pathway of paracetamol. The methylation reaction is catalyzed by the catechol O-methyltransferase enzyme, which is another important mechanism of rapid inactivation of endogenous and exogenous catecholamines. Drug metabolism can be affected by many factors including genetic polymorphisms of metabolic enzymes, age, gender, pregnancy, liver disease, and drug-drug interactions. The expression and function of phase 1 and phase 2 enzymes are reduced in neonates. The activities of some cytochrome P450 enzymes are increased in women compared to men. The concomitant administration of medications may also influence drug metabolism. The liver plays a very important role in all aspects of drug pharmacokinetics, including drug absorption, distribution, metabolism, and excretion. Venous blood from the gastrointestinal tract pools into the second capillary bed in the liver via the hepatic portal vein system. Some orally administered medications undergo extensive metabolism in the liver before entering the systemic circulation. This is referred to as the first-pass effect, and it may substantially decrease the bioavailability of these drugs. The liver also plays an important role in the distribution of drugs. Albumin and most other plasma proteins are produced in the liver. Impaired synthetic function of the liver may lead to a significant reduction of plasma albumin levels and altered protein binding of the drugs. Also, impaired liver function may lead to abnormal fluid distribution, ascites, and generalized edema that can further affect the distribution of fluids between compartments in the body and therefore can affect the distribution of drugs. We discussed earlier the role of the liver in drug metabolism. But the biotransformation of lipophilic drugs into more hydrophilic metabolites also facilitates their excretion in the kidneys. Furthermore, the liver is an important organ for the excretion of some drugs. Liver cells transfer various substances, including drugs, from plasma to bile by transport systems similar to those of the renal tubule. Various hydrophilic drug conjugates, particularly glucuronides, are concentrated in bile and delivered to the intestine, where the glucuronide can be hydrolyzed, regenerating active drugs. The free drug can then be reabsorbed, and the cycle repeated, a process referred to as enterohepatic circulation. Let's now discuss hepatic clearance. Hepatic clearance is the volume of blood perfusing the liver that is cleared of the drug per unit time. Hepatic clearance is determined by hepatic blood flow and hepatic extraction ratio. The hepatic extraction ratio is the proportion of a drug that is cleared from circulation during each pass of the blood through the liver. It can be calculated as the difference between the concentration of the drug in the blood entering and leaving the liver divided by the concentration of the drug in the blood leaving the liver. Hepatic extraction clearance is determined by hepatic internal clearance, that is the intrinsic ability of hepatocytes to metabolize the drug in the absence of other restrictive factors. Hepatic blood flow, and the fraction of the unbound drug in plasma, as only free, unbound fraction is metabolized. For drugs that are rapidly metabolized, in other words, drugs with very high internal hepatic clearance, the value of hepatic clearance approaches hepatic blood flow. Therefore, for these drugs, hepatic clearance is limited by the liver's blood flow. One such example is propranolol. This drug is mainly eliminated by the liver, and the intrinsic clearance is so rapid that hepatic clearance is limited by hepatic blood flow. Conversely, for drugs with low intrinsic clearance, the value of hepatic clearance is mainly limited to their plasma protein binding and intrinsic clearance of these drugs. The graph on the screen illustrates the relationship between hepatic clearance and hepatic blood flow for drugs with different values of intrinsic clearance. As you can see, for drugs with high hepatic clearance, the relationship between hepatic flow and hepatic clearance is almost direct, whilst clearance of drugs with low intrinsic clearance is less dependent on the blood flow.
The following questions on drug metabolism appeared in short answer questions in previous sittings of the primary exam. Outline the role of the liver in drug pharmacokinetics. This question appeared in the first sitting of 2016. 62% of candidates passed this question. The college's comment on this question was, most candidates structured their answer to this question well. They were aware of first-pass metabolism and the effect of protein synthesis upon the volume of distribution of drugs. Knowledge concerning phase 1 and phase 2 reactions was frequently inadequate. Many candidates were aware that these processes, as well as inactivating or activating drugs, resulted in increased water solubility to aid excretion via bile or urine. Few candidates discussed the significance of the large blood flow to the liver or the implications of high and low extraction ratios, especially in relation to liver blood flow. The next question appeared in the second sitting of 2016. Outline the anatomy and physiology of liver blood flow. Explain the changes to drug metabolism when liver blood flow decreases. 55% of candidates passed this question. The college's comment on the second part of this question concerning drug metabolism was, the question required recognition that hepatic clearance is the product of hepatic blood flow and the hepatic extraction ratio, and considering the impact on drugs with a high or a low extraction ratio. Many answers failed to adequately mention how hepatic blood flow was controlled, regulated, thus limiting the marks available. Similarly, in the second part of the question, many candidates spent considerable time mentioning the principles of drug metabolism rather than focusing on the question asked. The concept of hepatic drug clearance as the product of blood flow and its extraction ratio was poorly appreciated.